I'm going to quickly move us on to our next presentation. So next up, we have T-cell processing and immunogenicity predictions. You'll be hearing from Bjoern shortly again, but firstly, you'll be hearing from Austin Crinklaw. So Austin, I'd like to welcome you to the virtual stage. Take it away.
Hi, thanks, Nina. So yeah, I'm Austin. Let me share my screen. Sorry about that. So I was a research tech in Bjoern's lab for a little over 3 years. I mainly worked on creating new tools for the IEDB, but today I'm going to be talking about the T-cell processing and immunogenicity predictions. So just to give a walkthrough of what I'll be presenting on, I'm going to give kind of a broad overview of the biology and the motivation. I'm going to do a more specific look at some of the pathways, and then I'll be walking through some of the tools, and this is specifically for class 1. Then I'll pass it back to Bjoern, who will do class 2. So to begin, we're going to start with this mouse model, and we're going to have this virus, and this virus will infect the mouse, and then it might infect a cell, in this case, an antigen-presenting cell. And viruses, they spread, they replicate, and they infect other cells. And so kind of a fundamental idea here is the adaptive immune system. Now we can recognize if a cell is infected. So these viral proteins, just like other proteins in the cell, they're subject to degradation. So these small peptide fragments, they come from this virus. They can be bound to class 1 MHC molecules on the surface of the cell, where they can be recognized by a T cell receptor, in this case attached to a CD8 positive T cell. Once this occurs, it stimulates the immune response, and we could have cytotoxicity, so killing of the cell through the release of cytokines, or we could have a memory T cell response. The T cell could proliferate. So what we're fundamentally interested in here is if we have a set of viral proteins, which peptides in this, in this proteome are actually going to stimulate an immune response and undergo this pathway. So we'll take a closer look at the pathway. So say we begin with this protein. It's processed by the proteasome and cleaved into peptides. These peptides are generally cleaved further. They can then bind to TAP, or they're brought into the ER. They're generally cleaved again, and then they can bind to class 1 MHC molecules. These are then brought to the surface of the cell, where they can be recognized by the T cell receptor. So in essence, you have a pathway in which a protein is degraded, and the peptides from the protein are then eventually processed through this pathway and presented on the surface of the cell for recognition by the T cell. So these processes, though, they aren't random. They're specific. And when we're talking about the actual numbers of the peptides going through this pathway, so approximately 15% of all the possible peptides generated are actually going to bind to TAP and be brought in. About 2.5% of those are actually going to bind to the class 1 MHC molecule. And once that occurs and it's presented on the surface of the cell, then about half of them are actually going to be immunogenic. So now I'm going to walk through the tools. So the first one is going to be the class 1 combined predictor. So this combines predictions for the steps of proteasomal cleavage and DAP transport. And just to note, on the model itself, it was trained on specific in vitro data sets. So here you have your interface. You'll upload your protein sequences in FASTA format, and then we have some parameters that you can change. So we'll take a closer look at the parameters. So for the first one, we have the proteasomal cleavage prediction. So here, you can specify your proteasome type, so either immuno or constitutive. And just to note, the motifs that this tool is looking for, they were determined based on these proteasomal digests and the data from them. The next parameters that we have are the TAP transport predictions. So you have your max precursor extension and alpha factor. And so these are quite specific, and we suggest that if you don't really know how these are going to alter the predictions, that you keep these as default. So once again, here's your interface with the protein sequences uploaded in FASTA format. You would then hit submit, and here's what you get out. So you have this table here. This table is going to have information about what you uploaded, and it's also going to have the prediction scores. So looking at these closer, you have your proteasome score, TAP, MHC, processing, and total scores. So the higher the score, the higher efficiency for MHC class 1 presentation. And the MHC scores, these are negative because it's the negative log 10 of the IC50. And so these processing and total scores, these are the combined scores. These are additive. Processing is going to refer to the proteasome and the TAP score added together. And the total is the proteasome, TAP, and MHC score added together. And when you actually look at these scores, the variances across the scores are actually reflecting the selectivity in the steps earlier that I outlined. So we do need to talk about the caveats of the performance of these predictors. So generally speaking, the processing predictions, they're going to beat MHC binding predictions, but only when you're talking about eluded peptides. There's no evidence that these tools are better at predicting epitopes. The eluded peptides, they might overrepresent the best possible ligands, and this is kind of based on the idea that the MHC molecules have co-evolved to bind to peptides that are being produced by this pathway. So most of the high affinity MHC binders are also going to be efficiently processed. So our recommendation here is that you use the MHC binding predictions alone by default, but you might want to use this as a filtering step if you want to limit the number of candidate peptides. So moving on to the next set of tools that I'm going to talk about, it's the neural network-based predictors. So here we have three options. You have NetChop, NetCTL, and NetCTL-PAN. The NetChop, it's the proteasomal cleavage step. NetCTL is NetChop, TAP transport, and NetMHC. And then NetCTL-PAN is NetChop, TAP transport, and NetMHC-PAN. So the key difference here between these tools and the previous tools is the use of NetChop. And so here you could select your prediction method. I'm actually not going to be delving any further into these tools. So we believe in continuing to host these tools, but really it's kind of for archival or historical purposes. I'm going to just kind of gloss over these. The next tool I'll be talking about is MHC-NP. So this is the prediction of peptides naturally processed by the MHC. And so, like Bjoern mentioned earlier, we do like hosting other folks' tools, and this was developed outside of the IEDB. Here you can select your prediction method. In this interface, we have MHC-NP and NetMHC-PAN 4.0 EL. When these slides were made, this was the most up to date, but this is constantly evolving, and now we'd recommend NetMHC-PAN 4.1. And then for the final tool I'm going to talk about, we have the immunogenicity predictor. So the approach for this tool was to assemble two data sets of peptides. So the first set is going to be recognized by T cells. Well, they have similar MHC binding affinity. And the first set is going to be recognized by the T cells, and the second set is not. And when we looked at the peptides that are immunogenic, we see an enrichment of specific amino acids, but we also see a depletion of specific amino acids. So we use that information, the information about the enrichments, to calculate propensity scores. So here's an example. Here's the interface. You would enter your peptide sequences. Just to note, this tool, it was validated on 9-mer peptides, but you can make predictions for peptides of any length. Then we have your parameters that you can alter. So you have these mask positions, and the mask positions are just the MHC anchor positions. So you can either do default or your own custom list. Then you would hit submit. And here's what you get out. So you have this table, you have your peptides, their lengths, and their scores. The scores are just the sums of the propensity scores at all the unmasked positions. And if you have a high score, then it suggests that the peptide is more likely to be immunogenic. And then so looking at the performance of this tool, so like I mentioned earlier, experimentally it's been shown that about 50% of the MHC binding peptides can be immunogenic. When we cross-validated this tool, we had an AUC of 0.65, and when we tested on an independent blind set, we had an AUC of 0.69. And so looking at this graph here, we recommend that you use this cutoff of 0 if you want to have a high level of specificity. So I kind of blazed through this. So class 1 summary, just to reiterate, the processing predictions that I covered, they're better when you want to identify naturally processed ligands, but we don't have any evidence or data that shows that they can be superior when you're trying to identify epitopes when you compare to the MHC binding predictions. These specific predictions are good additional filters if your only goal is to select high likelihood T cell epitopes. And our recommendation is that you just use MHC binding scores and maybe consider pairing it with immunogenicity scores when you want to predict epitope candidates. And so that kind of concludes the class 1 portion. I'm going to pass it off back to Bjoern, who's going to cover class 2. And I'd like to thank everybody for their time.
Thanks, Austin. Actually, special thanks to you, because as Nina was saying, this is a guest appearance and probably the last one. Austin is free now. And it's really appreciated, Austin, that you step up here and give the presentation again. It's very, very helpful. So yeah, I'm going to talk about the CD4 T-cell epitope prediction part. And this is again about the processing and presentation predictions. And the same slide as I showed earlier, this is why class 2 is different. The pathway, how proteins get degraded and put onto MHC class 2 molecules is very different from class 1. And essentially in the processing and presentation tools, we're trying to predict these steps here beyond just the peptide MHC binding that contribute to what peptides are actually shown to a CD4 T cell and then potentially immunogenic. So let me go actually back here to my laser pointer. This is again how you get to the tool. Same thing as before under the T cell tools. You have two things that are relevant for a class 2 processing and presentation, immunogenicity, the MHC2-NP tool, and the CD4 T cell immunogenicity prediction tool. Starting with MHC2-NP, this is a tool where we looked for the motif of sequences around class 2 ligands. So given that, as I showed you, these are cleaved by proteases in specific compartments, and proteases always have a preferred specificity, we were wondering if can we find specific enrichment of amino acids at the ends of these ligands. So we used the ligand data that is derived from, that is found in the IEDB to do this analysis. And when we did that, we did indeed find very clear motifs, both at the C-terminal and the N-terminal end of peptides that are matching very specific cleavage motifs. And so we can use them to predict much better what ligands can be identified in a natural ligand elution experiment where you're identifying ligands found on the surface of of cells presented by MHC class 2. However, what these tools did not do, and this is the reason why we don't brag about them more, is improve T-cell epitope prediction. And this was to some extent a surprise. I'll talk about that in a bit later. So here, this is the tool entrance form where you put in the sequence. You don't actually have to add any other parameters, and you can just submit and then get the results back where the peptides are scanned, and you get back for each 15-mer peptide here, what is the peptide itself, and what is the N-terminal and the C-terminal motif. And these motifs are consisting of one residue outside of the peptide, so just before, and then the next two. And the same at the end, you have the two residues inside the peptide and then one outside, because those were the ones where we actually found a motif for the cleavage site to liberate these peptides. So we could generate cleavage probability scores here that then say these are actually the peptides that are most likely to be liberated just based on cleavage from the protein. Here is just the top 5. You can also get the complete results. And when we use this to identify what are the ultimately the predicted, what are the eluted ligands from MHC class 2 molecules naturally occurring in cells, we do actually have a pretty good performance of around 0.7 using just this cleavage score, which doesn't take into account the specific binding motif of an allele at all. We can combine that with the 7 allele score, which is able to, again, predict the likelihood of a peptide to be bound by these 7 alleles and then overall to be recognized. And then we can just say, okay, if we convert this, how likely is this peptide to be recognized by T cells? In this case, how likely is this peptide to be identified in a ligand elution assay? And you can see that the binding by itself and the cleavage by itself both work. And when we combine them using this simple linear combination, we have a maximum here of basically is using half and half of the cleavage score combined with the binding score. And then we have like a better combined prediction. Again, when we applied this to epitopes, it ends up not really working because for epitopes, as I showed you before, the ends of the peptides don't really matter that much. You can have longer and more extended versions. What the T cells see is ultimately the same, and these motifs are quite general, so they can be found everywhere. This is published in this paper, and independent of us, Morten Nielsen's group in Denmark actually had very similar results published around the same time. So this is this tool. Then the other tool we did is looking for immunogenicity predictions. So looking for motifs in peptides that are more likely to trigger T cell responses directly, and not trying to identify what actually the mechanism is, just training a neural network based on epitope data from the IEDB where we had overlapping peptide sets, and we looked for immunogenic peptides, and we tried to identify, is there something in addition to binding that distinguishes them? And for binding, we use the 7 allele method as our base threshold. So we trained a neural network here on these data sets, and then we validated based on tetramer from the IEDB, using actually a large number of studies from all over the world to compare these methods. This is how the tool looks like. Again, you insert your peptide sequence, and you should know this by now. You can choose the IEDB recommended, which in this case is combining the immunogenicity and the 7 allele method, and you're getting scores then back that combine these two. And when you look at the performance, this is, I think, the key slide. Again, we are able to get better performance by combining these two over the immunogenicity score alone or the binding score alone. But when you look at the scale here, we are really looking at a change from 0.705 to 0.72, which is not very impressive. And so this is one of the reasons why we haven't really been using this much in our own work, given that the difference here is rather marginally small, and we weren't really that happy with this. So summarizing this, similar to class 1, the enhancement of prediction efficacy is minor compared to using the MHC binding predictions. We can greatly improve the performance of identifying naturally eluted ligands, but that in itself is typically normally not that interesting. We want to normally get at epitopes. So as of now, we recommend to keep it simple and stick to the recommended MHC binding predictions, either for specific alleles using the consensus in that immunogen2 pan or the 7 allele method. And with that, I am done.
Thank you so much, Bjoern, again, and thank you, Austin, again, very special guest for today. So thank you both for that presentation. We already have some questions coming through. So at this time, I would like to welcome back Raphael, Austin, and Bjoern, and we'll kick it off with a few questions. So I think, Raphael, you presented first. Would you like to take the first question?
Yeah, absolutely. So from Taina, I noticed the reference allele sets available were generated based on publications from 2011 and 2012. Are there plans to update these sets with new data available at the allele frequency net database? No, there are no immediate plans, but that should make a difference. If you take a look at the allele frequency net database, even though the database has tripled, more than tripled in size, the additions since 2010 haven't changed what the most frequent alleles are. They've mostly added new alleles that are very infrequent, but the ones that we chose as a reference set haven't changed that much. So it shouldn't make a difference.
Thanks, Raphael. I'll take the next one, which is from Sivan, which asks, is there a list that shows how well the prediction algorithms can predict the peptide binding expression to HLA2? For example, how well is the consensus that can predict binding of peptides to HLA DR3, 4, or 5? So the two resources we have for that is the original benchmark method, which I think is Wang et al. 2007, where we had identified the consensus method. And then there is the live benchmarks that we're performing, where data like this is also available. I would caution against relying too much on allele-specific performance results, because very often the performance is highly impacted by the kind of test sets that we have available. And some alleles are studied in complicated questions and some are not. And that can greatly give you an idea of that things are much better, much worse. So I would much rather talk about the performance of class 2 predictions versus class 1 predictions overall than for specific alleles. If we had infinite amount of data available, we could make those statements, but right now I wouldn't be very overly confident with them. And that's what I have here. I think, Raphael, you have the next question.
I do. I'm interested, from Marcia, I'm interested in HLA 2407. I'm happy to see that there are now data in the IEDB for peptide binding to HLA 2407. Many of them from the eluted ligands, 2 from pathogens. So has the IEDB class 1 prediction tool been updated for better accuracy using these HLA 2407 data? I don't know. I'm not sure about this one. I checked the dates, and I would say, if I had to bet, I would say it hasn't. But I tested the prediction tool, and it found many epitopes in the example that I put there. But nevertheless, the NetMHC pen EL is trained on the elution data, and it is a pen method, meaning it transfers knowledge from one MHC to another. So I would be very confident in the result it gives. But I also checked the benchmark, and I have not found any results on HLA 2407 on the benchmark. So I can't give you a firm answer, but I think it's worth a shot because it is a pen method.
Thanks, Raphael. The next question is for me, I think. It says from Taina for Dr. Peters. As you mentioned, it's hard to know what DQA, DQB, or DPA, DPV pairs make sense from a physiological immunological point of view. Even though they are less expressed than the HLA-DRs, I would like to consider them. Do you have any general thoughts or insights on how to perform these alpha-beta combinations? Could you share some resources on these topics if they are available? But this is actually a really good question, and I would love to have a more confident answer in our own website. So there are definitely a significant number of combinations that are known to exist and make physiological sense. So essentially, if you look for alleles that are used in the IEDB, actually, where alpha and beta chains are specified for a significant fraction of epitopes, that is one way of identifying which actually make physiological sense, in that the hope is, I mean, definitely if somebody makes a tetramer, for instance, that you can actually pair those 2 chains. It would be awesome if somebody was doing like a comprehensive scan of which alpha chains can pair with which beta chains, but I'm not aware of that anybody has really done that. There should also be genetic, there's suspicions that, I mean, you co-inheriting your alpha and beta chains, that the ones that are co-inherited frequently together are the ones that are pairing together. But it is perfectly possible that you have matching of paternal and maternal alpha and beta chains in a mixed fashion. So this makes this more complicated. So yeah, so this is our current thoughts on the topic. Unfortunately, they're not as definitive as I would like them to be.
So I'll take Marcia's follow-up question. She wants a little more clarification on the second part, which is how about the immunogenicity prediction for peptides predicted to bind to A2407? I did test here the MHC1 prediction for using specifically A2407 for 9mers in an example, and it found many peptides that are now in the database. So the prediction tool worked very well. I can't say if it was because it had the examples included, or it is because it's a pen method that learns from other MHC examples. but the tool worked. I just tested it, and it found the epitopes that are now on the database, and it's below the threshold of 1% that I showed you on the presentation. So it worked well using the IEDB recommended method.
If I can jump in there, I think the question might be specifically for the immunogenicity part. So this would be for Austin. So immunogenicity predictions using the MHC1 T cell. And if I can just answer that, if that's the case. So those are completely universal. They are completely allele independent. And they're really just based on what residues are found more frequently, essentially facing the TCR and then are enriched in immunogenic peptide versus not. We have not found any allele-specific differences for that specific tool. And that doesn't mean that there aren't any. This might be a result of that we just don't have enough data to see differences in immunogenicity for specific alleles.
Excellent. Thank you so much. I think I can't see any other questions coming through. So I will thank Bjoern, Austin, and Raphael for the presentations and the times. Great. We had just one pop in. Thanks, Marcia. Awesome. So hopefully that answered a lot of your questions. Of course, there are multiple opportunities throughout the day for this as well. So thank you, Bjoern, Austin, and Raphael. I'll move to the next part, which is a short break. We will now, it will actually be supposed to be a 10-minute break. We've got about 15 minutes for a break to walk around, stretch our legs. So we will be reconvening at 3:45 p.m. Pacific time. So that's in 15 minutes. I will pop that in the chat though, so everyone has that to look back on. I'll also be sharing a link with some resources. Again, similar to yesterday, if you joined, there's some articles in there, some papers, links to our YouTube channel as well, where all of these videos will be posted for you. So feel free to browse that during the break. I'll also be resharing our IEDB handle for Twitter. We would continue to love to see all of your posts, so make sure you use the hashtag IEDBUW2021. I'll pop that in the chat as well as a reminder. But for now, we will take a break, 15 minutes, reconvene at 3:45 p.m. Pacific time. Alrighty, thank you everyone. See you in about 15 minutes.